Okay, and today what I'm going to do is I'm going to show you 7B2, and to do that I'm actually going to show you the last slide of 7B1. Remember, this is a codon wheel, and you can have a codon square. But basically what we do is we actually bring in 3 different nucleotides on the messenger RNA called a codon, and this is called a codon wheel. So we bring in like U, C, C, and we actually codes for an amino acid called serine. Then we bring in an AUG, which brings in methionine. This is actually the start codon. Remember, we do have stop codons as well. But we make these big chains of amino acids, and we can do it with the codon wheel. We can also do it with the codon chart. Hopefully we've done an activity to show that. And then B2 starts with this, and we're going to talk more about transcription and translation. We're going to break it down into steps. So step 1, DNA helicase, ACE. Remember, these are proteins, very special proteins called enzymes. And these enzymes do things faster at a different temperature than normal. And this is going to very quickly unzip and unwind the DNA. And then once that happens, step 2, we're going to bring in the 3'5 strand. Remember, we have a 3'5 strand, which is the lagging, and we have a 5' strand, which is the leading strand. We have to use the 3'5 because we want to make a 5' strand messenger RNA. And remember, the T's become A's, and the G's become C's, and the C's become G's, but the A's cannot become T's. It has to come up with a U, and that's why this U is going to this A. And that's the big difference between DNA and RNA. RNA has U's rather than T's. It does happen in an RNA polymerase. The DNA polymerase actually separates. The RNA polymerase is going to come in and actually create the messenger RNA. So it takes the RNA nucleotides, bonds them together, coding after the codons or the 3 sections of the DNA. And this is going to be read from what's called a gene on the 3'5 strand. And this gene may actually code for a protein for blood. Could be the color, could be the hemoglobin that's inside of it. We can get another gene over here. This is eye color. And we have another one over here, which is bone density. Different proteins do different things. Remember, you're created by proteins. Step 4 is going to be shown in an animation down here. mRNA leaves the nucleus. It now has a special code that actually makes a required or needed protein created by that gene on that DNA, and that protein code is going to leave the nucleus of the cell and actually find a ribosome. So here we go. We're going to finish it up with a stop protein, stop codon, and this is going to leave the nucleus. Then we're going to start translation. That's going to happen at the messenger RNA, where we bring in tRNA with their associated amino acids. They're going to get polypeptide bonded together. Here comes another one. Here comes another one, and it'll keep going until we get to the stop codon. And I'll let it go. I'll let it describe all this stuff when we get to translation. And here goes the stop. And then that protein will go out, and it, it will actually spin up like a ribbon, whatever shape it's supposed to be, and then go out and do whatever it's supposed to do. Okay, this is a shockwave file, which I'm going to click. It is not going to open up on apples. And then I'm going to push play, and I'll let him talk. Remember, we have a 3'5 strand, and we have a 5' strand, and we have a 3'5 strand. It's the 3'5 that we get it.
The synthesis of messenger RNA is called transcription. Transcription begins when RNA polymerase recognizes and binds to the promoter region on the double-stranded DNA molecule. A particular subunit of the messenger RNA, called the sigma factor, participates in recognition of the